Hi everyone. The paper we'll be discussing in our journal club this week is titled Enhancing Extracellular Vesicle Cargo Loading and Functional Delivery by Engineering Protein-Lipid Interactions, published in July 2024. A major challenge in EV-based drug delivery is ensuring that therapeutic proteins are efficiently loaded into EVs while still maintaining function. Current methods like post-harvest loading or genetic overexpression are often inefficient or disrupt EV integrity. EVs are lipid-based nanoparticles that facilitate intercellular communication. Their membranes are known to be enriched in lipid rafts, which are highly ordered microdomains that regulate protein trafficking. The authors hypothesized that designing proteins to associate with lipid rafts would enhance their EV loading. Their hypothesis is illustrated in Figure 1, showing that proteins that associate with lipid rafts in the plasma membrane are expected to be preferentially packaged into EVs. If this were true, it would provide a generalizable strategy for improving protein cargo loading into EVs. To test this hypothesis, the authors first conducted bioinformatic analysis to identify protein features that promote raft association. They found that proteins commonly found in EVs often possess longer transmembrane domains and specific lipid modifications. To validate this, they engineered variants of the transmembrane protein LAT, modifying its length and lipidation state. These variants were expressed in HEK293FT cells, which are a widely used human embryonic kidney cell line known for efficient protein production. Figure 3a summarizes the experimental workflow. Engineered proteins were expressed in cells and analyzed for membrane localization and raft association using microscopy and biochemical assays. Then, EVs were harvested, and protein cargo loading was quantified using SDS and fluorescence imaging. The results confirm that proteins with stronger raft association localized more effectively to the plasma membrane and were loaded into EVs at higher levels. Before applying lipid raft targeting strategies for cargo loading, the authors needed to confirm that EV membranes do naturally resemble lipid rafts. They first examined EV membrane organization using Largen spectroscopy, which quantifies membrane lipid order. Higher lipid order is common for lipid rafts, which contain tightly packed membranes. Figure 1c shows that EVs do exhibit high order, but this alone does not prove that EVs actively select raft-associated proteins. Next, the authors conducted bioinformatic analysis of known EV-associated proteins in Figure 1d to find that raft-associated proteins are significantly enriched in EVs compared to other membrane proteins. Finally, it was investigated whether cargo loaded through this method remains functional upon delivery to target cells. This was tested using synthetic transcription factors, or SynTFs, where lipid modifications were added to SynTFs to enhance their EV loading. To assess whether these EVs successfully delivered functional cargo, SynTF-loaded EVs were administered to their HEK-based reporter cell line, which fluoresces upon gene activation. Figure 7f shows that cells treated with lipidated SynTF-loaded EVs exhibited significantly higher reporter activation, with the best design showing a 240-fold increase in partitioning. In summary, the study demonstrates that targeting proteins to lipid rafts enhances their loading into EVs and improves their delivery to recipient cells. These findings provide new strategies for optimizing EV-based therapeutics by leveraging membrane biophysics to control cargo loading and delivery.